Imagine for a moment you're a tiny organism, navigating a world just teeming with microscopic life. How do you decide, like in an instant, which microbes are food, your friends, and which are pathogens? Dangerous. It's literally a matter of survival. Today, we're taking a deep dive into the incredible sensory world of Caenorhabditis elegans, this tiny roundworm, and specifically how it uses its sense of smell, or something like it, to make these lightning-fast, life-or-death decisions about what it eats.
Yeah, you've shared some really fascinating research with us. And our goal today, our mission for this deep dive is to unpack the neural circuits and the specific chemical signals that let C. elegans tell the difference. You know, how it distinguishes its nutritive microbial buddies from its pathogenic foes. Get ready, because there are definitely some surprises and aha moments in here.
Okay, let's get straight into it then. Our sources really emphasize that for C. elegans, there's just no time to sample something, get sick, and then learn it's bad. It has to know immediately. So how do they manage that? Such rapid discrimination.
Well, what's truly amazing here is just the speed of it. It's an innate ability. The research shows really clearly that C. elegans hermaphrodites, they gather very quickly, and they stay on lawns of nutritive E. coli. That's the good stuff. But they rapidly, like within minutes, avoid pathogenic Enterococcus faecalis.
Within minutes. Wow.
Yeah, exactly. This huge difference in behavior happens so fast, it strongly suggests it's driven by, immediate chemosensation, smelling or tasting it. It's not about waiting to feel the effects of an infection.
So it really is all about what they're sensing chemically. They can't rely on just bumping into things and learning that way.
Precisely. And the study gives some pretty compelling evidence for that. They looked at worms with mutations in key chemosensory genes, specifically TAX4 and OSM9. These are genes essential for chemosensory transduction, basically turning a chemical signal into a nerve signal. And when those genes are broken, the worms completely lose their ability to stay on the good bacteria.
Ah, okay. So that really pins it down to their sense of smell or taste, their chemosensory system.
Absolutely. It establishes right away that this nervous system input is central.
And didn't they even manage to see which specific detectors, which neurons in the worm's head were actually firing, like getting a peek inside its tiny brain while it smells?
Yes, they did. Using this high-sensitivity calcium indicator, GCAM-P6, think of it like a tiny light bulb inside the neuron that gets brighter when it's active. Really clever stuff. And they saw distinct populations of neurons responding differently. So neurons called ASE, AS, H, and ASK, they lit up preferentially when the worms were exposed to stuff from the pathogenic E. faecalis, like little danger signals.
Okay, the bad smell detectors.
Right. And by contrast, another said ADF, ASG, ASI, AWA, and even AWC neurons. These responded more to cues from the good nutritive E. coli. AWC is interesting. It often responds to the removal of a smell, but it was grouped with the good smell responders here.
So a really sophisticated setup, different neurons for good smells, different ones for bad smells, firing off right away.
Exactly. It shows this rapid, complex neural encoding of just what kind of microbe it is.
Okay, so you have these specialized sensory neurons sending out signals, danger or food. But how do those raw signals get translated into an actual behavioral decision, like move towards this or get away?
Yeah, that's the next crucial step, isn't it?
Yeah.
How does the nervous system process that information? In C. elegans, it's known that a relatively small number of interneurons act as kind of integration hubs. They get input from lots of different sensory cells. And the research points to the AIB and AIZ interneurons being really important for sensing microbes. AIBs in particular seem to act as a key bottleneck between the sensory input and the neurons that actually control movement, the premotor neurons.
Okay, AIBs are the decision makers. And this is where things got really interesting, maybe even a bit weird, right? The initial finding was quite counterintuitive.
It really was. Quite surprising. They use another tool, a calcium integrator called Campari. This lets you measure neural activity over time in worms that are just behaving normally, moving around freely. And they found that when worms were exposed to the good E. coli, the activity in these AIB neurons actually went down, a marked reduction.
Down, so they were inhibited.
Exactly. AIBs were inhibited by the smell of good food, but the pathogenic E. faecalis, it caused basically no change, or maybe a much, much smaller inhibition.
Wow, okay, so less activity in AIB means good food, go towards it. That kind of flips the script on how you'd think activation works.
It does, doesn't it?
Yeah.
It seems like inhibition of AIB is the key signal here for attraction, and the research backs this up strongly. They did experiments where they artificially silenced the AIB neurons. using something called chemogenetics, basically, a chemical switch, you say IL, to turn off just those neurons.
Like a remote control for the worm's brain cells?
Sort of, yeah. And when AIBs were silenced, the worms were much worse at navigating towards the E. coli. Their seeking behavior was significantly reduced.
So turning off AIB messes up their ability to find the food patch.
Right. But interestingly, silencing AIBs didn't really affect whether the worms stayed on the food patch once they got there.
Ah, okay. So maybe there's a different circuit for dwelling, for staying put.
That's what it suggests, yeah. AIB seems to be more about the journey to the food, the navigation part.
Like the GPS guiding you there, but not the comfy chair that makes you stay.
That's a good analogy. And they did further experiments using optogenetics flashing lights to control the neurons. Inhibiting AIBs with light pulses made the worms change direction more often, travel shorter distances before turning. This supports a model where AIBs help control reorientation during navigation. So the idea is, when a worm moving towards good E. coli senses the smell getting weaker, maybe it's moving away from the patch, the inhibition on AIB lessens, AIB becomes more active, and this triggers a turn.
A biased random walk, essentially. Turning more when the good smell fades.
Exactly. It helps them zero in on the source.
Yep.
So the level of inhibition in AIB neurons might effectively calculate how attractive or repulsive a smell is, guiding the foraging behavior.
Okay, that's a really neat mechanism. Inhibition as the go signal for finding food. So we know which neurons sense the good and bad stuff, and we know AIB inhibition guides them towards the good stuff. Now, the big reveal. What are the chemicals? What's the specific scent of good food that E. coli is giving off?
Right. This is where they brought in the heavy chemistry machinery. Untargeted liquid chromatography mass spectrometry, or LCMS. Basically, a way to analyze everything in the juice that the bacteria were grown in the conditioned media and see what molecules are there, comparing the E. coli juice to the E. faecalis juice.
Like a chemical fingerprint analysis. And what popped out from the good stuff, the E. coli?
Well, it was pretty striking. They found hundreds of metabolites enriched in the E. coli media, but 3 specific polyamines really stood out: cadaverine, putrescine, and spermidine, and their acetylated forms too.
Polyamines, like cadaverine. That sounds, well, not very appetizing usually.
Huh. Yes, the names are a bit grim. But N-acetylcadaverine, for instance, showed a huge enrichment, like a log 2-fold change over 7. That means it was over 100 times more abundant in the E. coli media compared to the pathogenic stuff. And further targeted analysis confirmed it. The E. coli media was just swimming in these polyamines, both acylated and non-acylated, a stark contrast to E. faecalis.
Okay, so E. coli makes a lot of these polyamines, but is there a clear link to the worm's behavior? Do these specific chemicals actually attract the worms?
Yes. They found a very clear correlation. They tested all panel of different bacteria, and the ones that caused that strong AIB inhibition we talked about, like E. coli, Serratia marcescens, Pseudomonas aeruginosa, all known to be attractive to the worms. Guess what? They also produce higher levels of cadaverine.
Ah, okay. And the unattractive ones?
Conversely, things like Staphylococcus aureus, which didn't inhibit AID and isn't attractive, very low cadaverine production.
So polyamines look like a really strong candidate for the come hither signal from good bacteria, the secret handshake.
They seem to be a major part of it, yeah. And they tested this directly. They made a synthetic blend of the main polyamines found in the E. coli media. And when they presented this blend, the ADF neurons responded to the polyamines appearing, and the AWC neurons responded when they were removed, exactly mirroring how they reacted to the actual E. coli soup.
Okay, so the sensory neurons recognize these specific chemicals.
They do. And the sensitivity, particularly of the AWC neurons to cadaverine, was just astonishing. They responded to concentrations as low as 1 femtomolar.
Femtomolar. That sounds incredibly tiny.
It is. It's like detecting a single drop in maybe 20 Olympic swimming pools worth of water. Unbelievably sensitive. And these tiny concentrations triggered responses just as large as much higher concentrations, like millimolar levels of the whole blend.
Wow. So AWC is exquisitely tuned to cadaverine.
It certainly seems so. ADF neurons responded to, but weren't quite as sensitive at the higher end.
All right, the neurons react. But do the polyamines actually make the worm move towards them like real food? Does it trigger that AIB inhibition?
Absolutely. Both the polyamine blend and, critically, cadaverine by itself were enough to cause that rapid inhibition of the AIB interneurons.
Okay, connects the chemical to the decision neuron.
Exactly. And behaviorally. Cadaverine alone was inherently attractive to C. elegans. They moved towards it. And crucially, if they silenced the AIB neurons using that chemogenetic switch,
The attraction should disappear.
completely abolished. which ties it all together beautifully. AIB inhibition, triggered by cadaverine, drives attraction. They even did an experiment where they took the normally repulsive E. faecalis media and spiked it with the polyamine mixture.
Did it make the bad stuff smell good?
It significantly improved the worm's chemotaxis towards it. They were much more willing to approach the bad smell when it had those good polyamines added.
That's fascinating. So it really seems like polyamines are a key part of the attractive signal. But does this mean it's only about polyamines? Is that the entire signal for good food?
That's a really important question. To test that, they did a clever experiment with a mutant E. coli strain. This strain had mutations in the genes needed to make polyamine, so it was polyamine deficient.
Okay, so if polyamines are everything, this mutant E. coli shouldn't be attractive at all.
Right, it's what you might predict. But what they found was that the attraction to the media from this mutant strain was significantly reduced compared to normal E. coli, but it wasn't gone.
So it was less attractive, but still somewhat attractive.
Exactly. The chemotaxis index, the measure of attraction, was still positive. This polyamine-deficient E. coli could still make some attractive cues.
So polyamines are important, maybe the main signal, but not the only signal.
Precisely. It suggests that while polyamines are a major component, C. elegans likely senses other things too. Maybe those ditin tripeptides that were also found enriched in the E. coli media play a role. It seems to be a more complex chemical language, a whole bouquet of smells, not just one single note that identifies good bacteria.
What an incredible deep dive. We've gone from behavior right down to specific molecules and neurons. So stepping back, what does this all mean in the bigger picture beyond these tiny worms?
Well, if you connect it outwards, it really highlights how important polyamines are as ecologically relevant smells across the animal kingdom. It's not just worms. They're known to trigger really diverse innate behaviors everywhere. Like invertebrates, mice, and zebrafish find them aversive. They signal danger or decay. But you have rats and fruit flies where they can be appetitive cues, signals for food.
And now we see clear attraction in C. elegans.
Right. And what's fascinating is that C. elegans seems to have evolved its own unique ways to detect them, different receptors and pathways than what you find in insects or vertebrates. It really underscores how fundamental polyamine sensing must have been shaped by evolution, specifically for these host microbe interactions.
It's amazing that such, chemically speaking, relatively simple molecules produced by microbes can act as such powerful good or bad signals across such different branches of life, a sort of universal microbial language interpreted differently.
Exactly. And it kind of leaves you with a thought to consider, doesn't it? If these tiny worms are using complex chemical cues like polyamines to constantly evaluate the quality of their microbial environment, well, how might similar, maybe undiscovered chemical sensing mechanisms influence our own interactions with the massive microbial world inside us and around us?
Especially since our own gut microbes produce polyamines too.
Precisely. And we know polyamines can modulate things in our own bodies. So are we subconsciously sensing microbial signals in ways we don't even realize yet? There's definitely always more to learn.
A really great thought to chew on after this deep dive. Thanks for unpacking that world for us.